I'm oral historian Mike Chappell. Today, March 22nd, 2010, I'm interviewing Dr. Jerome Hershman in his office at the West Los Angeles VA Medical Center. Dr. Hershman has made important research contributions to a wide variety of topics in the field of thyroidology. He pioneered the interrelationships of thyrotropin, chorionic gonadotropin, and their receptors, and he was among the first to point out the effects of vasopressin, tumor necrosis factor, transforming growth factor, thyrotropin-releasing hormone, and cytokines on thyroid function. More recently, Dr. Hershman's research has contributed to our understanding of the mechanisms of formation and growth of thyroid tumors and the role of the sodium iodide symporter. Dr. Hershman is past president of the American Thyroid Association and the founding editor of the journal Thyroid. Currently, Dr. Hershman is Associate Chief of the Endocrinology and Diabetes Division of the West Los Angeles VA Medical Center, as well as Distinguished Professor of Medicine at the University of California, Los Angeles.
I was recruited for the position at UCLA, really at this VA hospital, West Los Angeles VA, known then as the Wadsworth VA Hospital, to run the division here, really to get it going again because it had fallen into disuse. The previous head had left 2 years before, had not been replaced, and they were looking for somebody. The chairman of medicine at UCLA at that time in 1972 was David Solomon, who had actually worked in Astwood's lab, was an endocrinologist of excellent stature, and so he recruited me along with the chief of medicine, Seymour Dayton. They brought in a diabetologist because we have a lot of patients with diabetes, Seymour Levin, who has remained a lifelong colleague, and then I brought in the following year another person, Harold Carlson, whose expertise was in the pituitary, and we collaborated together to do a lot of work, again, TRH related. I developed, with the help of Henry Friesen, who had been a fellow with me at Astwood's lab, who had developed, who had purified prolactin, developed an antibody to it. So I set up an assay for prolactin in my laboratory here. I had a very good TSH assay that I'd worked on, but I set up the assay for prolactin, and we and others showed that TRH caused a rise in prolactin, and that was work that Harold Carlson did because he was interested in prolactin. So we were a nice team. He worked on prolactin, I worked on TSH, and we did a lot of work in neuroendocrinology at that time in that respect. I decided to move in the area of thyroid cancer. The work that I became interested in doing was mainly at a basic level, because even though we have a lot of thyroid cancer here, the populations that we have to follow are much smaller than those of big cancer centers. So I decided to focus more on the basic science of thyroid cancer, and I was fortunate to have two excellent molecular biologists who joined me. One of them, Michael Fenton, is continuing to work with me. And I was also interested in the sodium iodide symporter, which Nancy Carrasco cloned now almost 15 years ago. And we showed that we found, as did others, that the symporter was relatively inactive in thyroid cancer cells, and we decided to work on trying to improve that, as have other groups, trying to activate it so that thyroid cancers, papillary cancers, could be treated with radioiodine. So in order to improve it, you have to find out exactly why it's not working. Well, this led to studies on the molecular regulation of the sodium iodide symporter, and we studied the far upstream enhancer. A Japanese postdoc came to my lab and worked on this and found that there was a region in what was called a far upstream enhancer of the gene that had some regulatory components, and we found that a binding factor, which was necessary to activate it, bound to this upstream enhancer, and it would activate the symporter, was deficient in a line of papillary thyroid cancer cells. And Mike Fenton, who joined me subsequently, showed that the missing factor was a transcription factor called CREM, C-R-E-M, which has a lot of other roles in the cells of the body. And so that was the main contribution in recent years of our work on thyroid cancer, was really on the regulation of the sodium iodide symporter, and I've continued to focus on that. Well, the field of endocrinology has enlarged a great deal since I went into it when it was more physiologically oriented. And now the emphasis is on molecular biology on the one hand and on evidence-based medicine on the other hand in regard to clinical applications. And I think there's probably some more separation between clinical endocrinology and basic science and endocrinology. Fortunately, the Endocrine Society has an annual meeting at which both are presented. I think that it's still a wonderful field with a lot of challenges. We have a large number of applicants for fellowship, mainly wanting clinical training, but research training as well. And so I think the field has done very well and will continue to change in ways that are probably completely unpredictable at the present time.